you. So thank you to the organizers in IOC for, organizing this great workshop. The morning was fantastic, and I'm very honored and grateful that you asked me to come here and talk a little bit about what we are doing in the lab. And I wanted to, because it's an education workshop, and I know there are quite a few people on Zoom as well, I want to really tell you a little bit about the lessons we learned the hard way, what worked for us, what didn't work for us, and how we are trying to really move from bench to bedside. So, if I can... No. So, we are working in the oncology sector. So, most of our work is in cancer. And really what we are basing our work on is really the key clinical problems. Half of my lab is working on cell and mouse models. The other half is working really with patient material, interfacing with the clinicians. So we really went to the clinicians and said, what are key clinical problems, for example, for this non-small cell lung cancer patient here? And they said, well, the key problems are obviously patient management. So what is the best therapy? What is the most promising one? But also the diagnosis. The early diagnosis. So I will touch on the early diagnosis a little bit today, just as a red line to go through some of the isolation protocols and what we have been doing. So how do we translate bench to bedside? Now, my group, we started long time ago in the exosome field and then shifted to the EV field. So there are a couple of the Jeff papers we published along the way. So we really from the beginning wanted to optimize the EV isolation, the exosome isolation. So the first paper where we were allowed to use exosomes in the title, back then in JEV, from 2015 has over 1,600 citations. And we really explain and analyze different ways to isolate EVs from cell culture as well as from human plasma. And then more recently, a few other isolation protocols. But here, for blood EVs, we have really used size exclusion chromatography, and we found that we can actually isolate very pure EVs. So we have used a lot of these for omics, or RNA-seq, proteomics, you name it, but also for the biology, where we wanted to exclude as many of the contaminants we have discussed this morning about. However, it's time intense, so 24 to 40 if I really ask for a lot per day per operator, and it's costly, or minimally costly, but I'll come back to this later. So what we have done over the years is found that, for example, by proteomic analysis, if you use cancer cells with different mutation patterns, what we know now is that part of the content of the EVs is depending on the cell of origin, so you get different cell of content of the EVs based on the cancer mutation pattern of the cells. So I told you I want to really take this as a highlight, the poor outcome of this gentleman who was part of one of our clinical trials with a non-small cell lung cancer. Because we diagnosed him that day, the chance of a poor outcome is much higher. We haven't invented a time machine, unfortunately, yet, but if we could transport him back into the past, diagnose him earlier, the chance of a good outcome would be much higher. So therefore, can we find cancer in this asymptomatic population of people who have small cancers, and most importantly, cancers which will eventually grow and potentially cause morbidity and mortality in these patients. So can we develop a cancer biomarker? As I said, we have done a lot of proteomics on cell lines and the cancer content or the cancer cell-derived content of EVs. So here we actually transfer these to untransformed and oncogenically transformed cells and look at the EV content. So here, human bronchial epithelial cells, which are isogenic cell lines. So we take an untransformed lung epithelial cells, we introduce a variety of oncogenes. Here are a few, we have several of these isogenic lines, and we do proteomic profiles, RNA profiles, but for today I stay with the proteomic profiles. What we can find is that there are quite a number of proteins which are different between the untransformed and transformed EVs. listed here in the heat map by proteomics. 4 of them, actually a few more, but here I'm showing 4, we have actually validated by ELISAs. So you see that there's an increase in the protein amount in the EVs. And then we went through different cancer cell lines, always trying to compare it back to normal or untransformed cells for lung cancer, brain cancer, colorectal cancer, breast cancer, and so on. And you see a lot of red down there, means there's a lot more of these 4 proteins in the EVs from cancer cell lines. So what is the clinical utility? And this is really where we are facing the problem of EV isolation, bench versus bedside. So here, within the first cohort, before the reviewers told us to do more, we isolated the EVs from blood of 250 healthy age and sex-matched subjects and around 500 cancer patients. Thanks to reviewer questions, we have increased these now to close to 1,000. And what you can see here are in green, the healthy, and then different cancer cohorts and the amounts of this one biomarker protein. And you can see that there's a trend, for example, in red, the non-small cell lung cancer. You can see that this is higher in general, but we have quite a few in the healthy samples, which are higher than in the cancer samples. So one biomarker is insufficient to actually give us information. So then we went back, and I have to explain a little bit here. We have done this for the 4 biomarkers. We have done this independently by 2 of my people in the lab, so blinded, they didn't know that they are working on the same sample. And the data was all analyzed by biostatisticians, and the bioinformatician did machine learning to come up with an algorithm. This means that for each of the samples, we used around 1 ml to 1.5 ml plasma as input material to actually get all this work done. The good thing is that we have a very accurate discriminator between cancer and normal. Here are the areas under the curve for the 8 different cancer types. And if we go and look at different specificity cutoffs, the sensitivity is quite high, above 80% for most of them. Small cell lung cancer and prostate cancer are outliers. We had only a few samples out of collection reasons for small cell lung cancer and prostate cancer because it's not very useful. For 3 of the cohorts, we had enough early stage cancer. Remember, we want to detect cancer early. And there, if we look at stage 1, so the earliest of the cancer stages versus the other cancer stages, you see that the sensitivity at the different specificity cutoffs is very similar, meaning that we can actually detect cancer, non-small cell lung cancer or gastric cancer or esophageal cancer at the earliest stage in the blood of individuals. So quite accurately. But I want to come back to this because that is really our big problem. So I told you before, the first detection cohort was around 1,000 patients. Over 1 ml of plasma means we had to run 2 size exclusion columns. We started it this way, so we don't change our SOP halfway through. So we had to do everything there in these. So these are already 2,000 samples we isolated. We have then a prospective clinical trial. So overall, I discussed with the lead postdoc, we have just for data, which is included in the manuscript at the moment, we have done over 4,000 extractions of EVs. Now, I told you, if I really force it, if we're really urgently, we can do 40 samples per day per operator. We have done over 4,000, and it is where all my time went from the lab for the last few years. So we have done a lot of these isolations. So that is obviously great, but it's not very applicable for the clinic. Also, the money for the column is quite cheap. But if you think that for 40 samples you need a postdoc or a research assistant or anything to work full-time, and it's more or less a full-time job for the whole day to isolate the samples, the costs get actually higher. So we thought about other ways. One was, as part of this project, we collaborated with Matt Trow from RBN at the University of Queensland. RBN is the Australian Institute of Biotechnology and Nanotechnology. So we wanted to see whether we can avoid the plasma isolation. So as Rink has started and the whole morning discussed, there's difference between plasma from one source and another source. And what we came up with is a little microfluidics device, so nanoshearing, where we can actually have a capture antibody. We induce with AC current a little flow, so this helps the mixing and prevents unspecific binding. And then we can have these capture antibodies for the EVs. And then we have SIRS nanotags, so surface enhanced Raman spectrometry, which are coupled to antibodies, and therefore we get a signal. So here we have done this for CD63, CD9, CD81 when we set up this way of detection. And this is a false colored image. Each of these dots, if it's only red, green, or blue, is one of the markers, so only CD9 or only CD63. And if there are two colors overlaying, we actually have an EV which has both of the markers on top of them. So this is great because we can actually use blood instead of an EV isolation. There are some technical issues, but you can actually do it. And I show you only a tiny bit of the data here. So we actually were able to transfer these to our biomarker study. Here were the first 6 samples of healthy and non-small cell lung cancer for our biomarkers, so not for the tetraspanins. You see just by eye that there are many more signals for our biomarkers in the cancer samples. So this shows, and this was raised in the morning, that our test, our readout is robust. It's platform independent and also laboratory independent because we just gave the samples to our collaborators who did everything, not knowing whether it's cancer or not. So we did this and then expanded the whole study to the prospective clinical trial. However, doing it, we realized this is a great laboratory way of doing it. We were very hopeful in fact in the beginning because you need tiny amounts. You can go straight from the blood, but the nanoshearing combined with SIRS detection advantage is, yes, we don't need EV isolation, so this is cutting out a problem, but it's very time and labor intense. It's extremely low throughput, not like an ELISA where you can have a 96 well plate. So this is a very much sample by sample processing or 8-plexed samples. There are batch effects for the SIRS probes. Everyone who has worked with SIRS knows that there's a little batch effect. So we had to do a lot of normalization, which is again taking time and labor into account. And you needed very, very specific equipment, which no one of us or most of us will not have, and certainly not labs which could do cancer testing in our case. And it's therefore also very expensive. So also not a great idea. Now, the last part I want to really go into detail about is HPLC. So a few years ago, I have to say I moved a year and a half ago from Brisbane in Queensland, Australia, to Chinese University in Hong Kong. And in Brisbane, we collaborated with Matt Trau, but also with Carlos Salomon. And we discussed why not we use an HPLC. HPLC has been around since many, many years. I saw HPLCs during my studies, and they were last millennia, so it's a long time ago. And so why not use HPLC? We find these machines quite often in pathology labs. So it's translation ready. We don't need to buy specific equipment or anything. It's automatable, and I will come back to this. It's reducing human error. We can configure the columns for different requirements. And we have played around initially with Carlos. This is a machine in Hong Kong, our machine. So we can vary the volume, the throughput, if we want to do a lot of small volume or very pure of little sample number. And we can change the chemistry, so size exclusion, ion exchange, chromatography, affinity, and. So we could even precipitate or retain specific antibody-mediated EVs. So with this in mind, and Carlos is happy to share our data here and related this work, really we collected these around 180 samples of 5 different cancers and benigns and normals as controls. These are blood samples collected from different sites, plasma samples. And here we look at the protein profiles of normal versus cancer or metastasis for breast cancer metastasis. And you see we get a really nice separation of the proteome profile between cancer and normal. And this was done really as a proof of principle. For example, the lung cancer, we had our biomarkers. Ovarian cancer, Carlos had biomarkers, and a few other collaborators had their biomarkers for their cancer type. So we actually did the isolation and then confirmed, for example, our 4 biomarkers I showed you before are in the lung cancer proteomic profile and actually quite nice standouts. So what we are doing in the lab at the moment to really finish this off is we have a pipetting robot. So on the left-hand side for you, this is a demo at the moment. We will get a little bit bigger one where we will really aliquot the plasma samples into the different well plates. Then one person has to change it, and they're standing back to back. Take the plasma, put it into the HPLC, and make sure the buffers are there. And then the HPLC is isolating. At the moment, we use 100 microliter plasma per isolation in 96-well format. And then later on, we can actually take these 96-well plates out, put it back into the robot to pipette our ELISA plates out. So we don't have a lot of human interference, so the error rate is greatly reduced. We have done a few tests, and although I've got the perfect lab, but we're all humans, a robot is not making these mistakes because we didn't have coffee. So we are at the moment setting this up in the lab to see whether we can actually get this in a higher throughput. So at the moment, we have only one HPLC column in our HPLC, and it's 288 samples, so 396-well plates per day. And operator time is maybe 30 minutes, topping up the buffers and moving the plates back and forth. We can actually put 6 of the columns in there, so we would have 6 times the throughput if we wanted to. And therefore, it's quite cheap. The HPLC columns are regenerated, and Carlos has one which is several years old, and it's still working quite nicely. So that is one of the ways forward, how we are actually trying to translate this into the bedside or into a commercial setting. And it was one of the key reasons for me when I considered moving to Hong Kong or not, because there are actually pathways, a little bit more likely pathways than Australia to actually try these in a commercial or semi-commercial setting. Now, I want to finish this off, and we do a lot of this development in the lab, but as soon as you actually go into this kind of development, you will have a lot of interface with companies. And one of the companies approached us whether we can actually help developing an immune affinity device. So similar to the corona tests we all learned to love a few years ago, for low sensitivity, low specificity, black and white detection. And it's a point of care device that nurses or doctors could load the blood, and we can actually detect our EV marker or the company's EV markers there. So there are different ways. And as take-home message, as summary, I thought in preparation for here, and I knew Jody would give us a more generic overview and review, so I thought I'd give you a few lessons we learned, because there are so many pathways forward. I thought for us it was really important that we define the purpose of the EV isolation. So do we want the highest purity, or are we happy with contamination? Is it the time? Is it the cost? What is really triggering why you do the EV isolation? So there's no one size fits all. Are these EV isolation and later on the application, for example, our biomarker test, is it suitable for a transfer to the bedside? So we did the isolation many years ago when EVs were called exosomes and we didn't even know anything about it. So we started very, very early and we just wanted pure EVs. So that was great for this purpose, but not for the bedside. So with, benefit of hindsight, obviously I would have changed the direction back then if we wanted to end up at the bedside. Will it be used in a general or specialized lab, pathology lab? We are an international society, so each country, each region has different requirements, how you actually translate these into practice. Is it a point of care device, as I just mentioned, we are developing, or can you do it in your lab? Is there enough throughput? What are the equipment requirements? For example, STIRS, which is $1 million outlay and very specialized for this one application, with HPLC, which will stand around in pretty much every second lab or institute. And then the technological complexity. I'm a very simple person, I'm a very dumb person, so for us the more simple it is, the better, because the more complexity you build in, you might get better data, but it's getting much harder to do, to collaborate if you need special requirements. What is the industry uptake? That is something I'm having a very steep learning curve over the last couple of years, how the industry sees your test versus how you see it. And a big part there is the economics. They want to see how much does it cost, how much could it generate, how quickly can you do it, is it suitable for pathology, for the testing. Now with this, I want to think, and this is the collaboration, these are just the collaborators we worked with on the biomarker test, on this test I showed you here, as well as Carlos for the HPLC. And my lab, I was extremely lucky and I'm very, very honored that I was able to get all my lab transferred from Australia to Hong Kong. You see on the left-hand side, we needed much more protection in Australia, and we are now looking much more serious in Hong Kong. The only person we lost is Sophie, our research assistant, who stayed. The rest has, in the meantime, all moved back, and we were able to recruit 2 new students to the lab, and we are continuing our work there. Thank you very much.
Thanks very much, Andreas. I think it's a great talk. It's very insightful, lessons learned. So are there any questions? Yes, in the back.
Thank you very much. My name is Jill. I was wondering, I really like your workflow. And are you also considering to automatize the plasma preparation, or is it already automatized?
Yes, so for our prospective clinical trials, if we have the authority to do so, automation, yes, we first were actually writing SOPs, which are applicable and transferable. So we have done SOP process as, at least in Australia back then, every clinician would do al gusto. If it's a surgeon, it would take a little bit longer. A blood service would do it faster. So we have written an SOP to actually automate or to normalize it. And yes, we would like to automate it, but because of the centrifugation steps and the manual handling in between, we haven't found a way to automate it in this way. And coming from the other side, if we are looking at rolling it out across the country, across different pathology labs, if it's not a central lab, everyone is producing the plasma a little bit different. And that is going to the robustness of the test, which I haven't touched here because it's not the biomarker work, but we made sure that our test is robust enough that if we give you the samples or ask you to do the samples, we have done this with collaborators, where we said, here's the ELISA plate, we pre-coated it, do it this way, that you get very, very similar results from 200 samples you're producing to 200 samples Judy is producing in Hong Kong. That this is so robust that it doesn't, I understand, and Rink and I had discussions about this at several occasions about the plasma and the plasma input, but we have normalizations inside the test to normalize it to particles, to abundant markers, which were a little bit harder to define, that we can actually say, well, there's an increase.
Okay, thank you.
Andreas, just a quick question from the chat. What briefly the operation principle of the HPLC for detecting cancer-specific EVs in the blood samples?
So at the moment, what we are doing is a size exclusion column. So we are running a size exclusion column similar to the manual ones, just a very slim and long one. So, and then we get the EV isolation in a 96 well format and run our ELISAs. So We tried different detection methods for the markers, but ELISA seems to be the most robust, reproducible, also across different labs, and you don't need special equipment. So the HPLC is really EV isolation, and we are mostly using size exclusion here.